Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
So good morning and welcome to the final session in the Health and Wellbeing track at the Science Workshop. It's my pleasure to introduce to you Chen from the Purdue University Department of Computer and Information Science. And Jake will be talking to us today about proteome-scale protein isoform characterization with high-performance computing.
Well, thank you very much, Simon, and thank you for coming this early and to my talk. I thought I would have known everybody up until 2 minutes ago because I know May is a very good colleague and Jeffrey, of course. And so we have a small audience, and then I think we also have plenty of time. So feel free to interrupt me. And I think I'm giving this talk here is really I'm emphasizing on the science part. And I'm a bioinformatician and far away from a specialist in high-performance computing. But here I want to pitch to you that there's a need for large-scale science, especially in proteomics and through this case study. And then maybe if you're a person who really specializes in high-performance computing, take this problem and see if you can help us, right? So a little bit of a background, and I think for those of you who know that there's a lot of advances in the human genome sciences, especially with the sequencing of human genome about almost 9 years ago, you know already that there are lots of genomes, and more than 1,000 genomes has been sequenced today. But what I want to remind you is that today the genome is not the only thing that we're trying to study in bioinformatics or post-genome biology. If you follow this central dogma that the genetic information flows from DNA to RNA to proteins and eventually get expressed into a phenotype, and then we're actually studying multiple omics. At the transcriptome level, transcript level, we study the transcriptomics or functional genomics. At the protein level, we study the collections of proteins and their interactions. And we also try to capture all the phenotype together into a phenomics. And overall, the end goal is to actually study all the omics and in parallel and to measure the very complex different aspects of the biological system to study the system's biology. And of course, if we the end goal of systems biology is that we can do this science instead of doing the perturbations in a experimental platform, but then we can transfer gradually into an in silico platform so we can do all kinds of predictions and simulations. And why do we study, I think, proteomics? Why do we think that proteomics is a major study? And we all know that this caterpillar and butterflies, they are actually coming from one single genome, but what the life form shows is quite distinct. So this is one distinct example of one genome can have two distinct proteome. In the case of humans, and when you think about it, we actually have different type of proteome inside our body. If you take a look at the different type of tissue and cells, like ranging from skin cells to brain cells and the muscles and so on and so forth, we have a dramatically different type of the phenotype demonstrated in the cells. And the cells are nothing more than the collections of the systems that eventually made up of proteins and metabolites. And they're all actually starting from the same genome. And that really outlines the emerging needs of the study in the proteome. Right? It's much more complex than the genome, but we must study it in order to understand the complexity of life. Well, there are lots of challenges for the study of the proteome, especially for human. And one of that is that the proteome is not steady. It's very context specific, and it depending on what time you look at it, right? We can say we are studying, let's say, the proteome, all the proteins in our blood, depending on whether you eat breakfast or not. And I think that there are different liver enzymes that actually get eluted into the blood, and then they're different. And then also depending on many other factors, your age or health, tissue type, or environmental stimuli, for example, taking drugs or not. And then the protein may be different. And they fluctuate depending on the different physiological conditions, and sometimes it could be a disease. So it changes, but it's for both good and bad. The good thing is that you can use the proteome for some kind of diagnostic applications. And also, the proteome is not steady. And the genome, everybody for more or less the same genes or different genes, the variations of the copy number may not be that dramatic, right? So it could be 1 copy or maybe 10, 20 copies maximum. But here, we're talking about the proteins that can vary in concentration up to 10 to the 10th power. Something like albumin that exists in grams per mil in our blood, and then some cytokines that exist in just nanograms. And also, we have an enormous diversity of the protein isoform. And it's mostly because of the complex mechanism involved in just modifying the initial genes into the gene products. For example, splice variants, post-translational modification, and genetic polymorphism. And in human, we know that we only have about 24,000 genes, but when you take into consideration of all of those isoforms, and then we can have up to 1 million copies of the isoform. And they're also very tricky to study. And today, we just had a conversation with Paul, our next speaker, and our instruments are not very accurate in measuring this. Unlike those the genome sequencers, we can actually accurately read out the sequences up to maybe 5 sigma degree of accuracy. But for the mass spectrometers, and they're fairly inaccurate, and we have a tremendous up to 30% of false negatives. Even detecting them, we have problem, not alone with the bioinformatics of analyzing them. So there are lots of challenges, but I think the right now, the standard way of doing the proteomics analysis is something like this. You have a complex sample, a biological sample with lots of proteins, and then you usually, in order to reduce the complexity, you use some enzymes to digest the proteins into peptides, into a complex mixture. But it still is very complex, so you need to actually apply some protein biochemistry to separate the peptides according to the charges, and so that they are well separated. And then for each separated fraction, then you subject it into the mass spectrometer. So usually it's a Tandem mass spectrometer, so that you can also not only separate out the peak and get the MZ ratio, but also the peptide sequences. And after that, you just search for databases, known databases. There's a part missing here, but eventually you get the protein identification and the abundance information. This is a process that usually is used. However, I reminded you earlier about the complexity of the proteins, and the central dogma that we spelled out earlier has to be modified. And for example, this DNA in the process of becoming a messenger RNA. There are different level of modification and regulations, and most importantly, there's alternative splicing. As mRNA make its way into the proteins, there are different type of regulatory mechanism, but there are different kind of editing and post-translational modifications that's taking place. So therefore, we have this level of complexity. Well, in proteomics, and I think most people tend to make a very straightforward analysis and saying, well, maybe from genes to proteins is roughly 1-to-1. But that's obviously it's not the truth for biology. So in order to study a much deeper level of biology, we need to actually give a higher demand for proteomic studies so that we need to study those isoforms. Right? I think what an isoform is just there's any of the several different forms of proteins derived from the same gene. And a protein isoform, as we mentioned earlier, they're very essential force of achieving molecular diversity in eukaryotic cells. And so obviously they're very important for mechanistic studies or even the diagnostic applications. And because their importance in being associated with diseases such as cancer, and so they can also be used for disease biomarker studies. One of the common tests called a PSA test, maybe many of you know, is actually used for testing prostate cancer. But I think the PSA test initially was developed as a test and that suffers from loss of the false positives. And later on, people find out that actually when you measure only the total PSA, which is a protein, you're actually ignoring the fact that there are different isoforms, and some of the isoforms are actually naturally occurring in healthy individuals, and there is only the malignant part of the isoform that are strongly associated with the prostate cancer. So later on, they modified the PSA test to measure the different ratios of the malignant cancerous isoform versus the total PSA, and then they actually dramatically increased the specificity of this test. So as you know, the isoforms are very important, and so are lots of other proteins like cancer-associated pCNA, which is a protein involved in DNA replication. And there are different ways of thinking about the protein isoform. So this is one example is coming from alternative splicing. Let's say this gene has 5 different exons, and after alternative splicing, maybe the cell decided that, well, this is a mRNA transcript that would contain each one of the exons, 1 through 5, and therefore you get the protein A, that is the whole protein. And then in this specific form, exon 3 is called splice out. So you skip the exon of 3, and then you actually have a different protein that lack a specific intracellular domain. Or in this case, you have exon 4 splice out, and therefore you actually lack the transmembrane domain for the proteins. So the alternative splicing is one thing that we like to study in the protein isoform study. Another way of having the isoform is the genetic polymorphism. For example, in this case, we're seeing a single nucleotide polymorphism found in a particular gene, PRB gene. And this site originally, most common form is A, and then it could also be G. And when it's changed to G, then the translated amino acid corresponding to this protein goes from tryptophan to alanine. So there's obviously changes and consequences at the protein level. So we like to study all of these changes, which actually has not been incorporated in the standard proteomics analysis. And so here's the eScience part of it. And of course, we can't do anything about the experimental procedure, but once you have the captured proteomics spectrum data, which contains all the potential variations of the protein isoform, And I think there are different tasks. And for example, you need to convert the raw data to a more open format so that we can search it using the AMSA, something like that, and to search against the database of known proteins. And then you need to identify peptides and do those conversions. And a key to this process is that what database you're using for searching the mass spectrometry data. And if you look at all of the major databases, even though they may have very good number of entries that correspond to a large number of mRNA transcripts, but they actually have very poor representation of protein isoforms. And that's not what these databases are meant for. And especially for this alternative splicing transcript database for human, it's only got about 7,000 isoforms. And that's obviously not enough. So what we did in the previous work was to say, well, if this is the standard approach, you take the mass spectrometry data, search through using a AMSA database, and plug in a standard protein sequence database to get the protein identification information. So why not just have a new platform? And this new platform will take all of the gene information and the structures of the genes and the exons and introns, all the SNP information from DB SNP database, and then we generate a hypothetical set of peptides that correspond to different exon junction or exons or intron regions with or without a SNP. And then we create a database called PEPPI Database, a peptidomic database of human protein isoform. And then we use this database to be searched against using this software. Then maybe we can actually find more information from this mass spectrometry data. And that's the basic idea. So this database was published previously. And this database actually captures all the theoretically possible configurations of the protein isoform. For example, if you don't think about genes as a single continuous region, you think about genes as a combination of exons and introns, then you can actually capture the type 1 peptide as a single exon, or type 2 as normal exon-exon junction regions. Right? You should have a peptide that covers these regions, or peptide cover the regions that with any number of exon skipping events, or that in some rare cases you may actually have certain introns retained with the exons or intron actually just immediately prior to the exons, or partial introns as long as it doesn't contain the stop codon. So we consider all of these possible configurations in building the PEPPI database. And so as you can see that we map out this all of these regions to the original gene coordinate and call it exon, exon-exon junction, exon-intron, intron-exon, and this EETH really means the theoretical cases. So we want to make sure that we can potentially capture novel information by having this all the theoretical configurations in the search database, so that we may find novel information. And this is what the statistics of the PEPPI database is. So there are about 23,000 coding genes and 150K SNPs for the human. And then we captures about 200,000 exonic regions and exon-exon combinations, 200,000. And this with intron combinations, another 500,000. And hypothetical combinations, because they're there are a lot more cases, and so you get to up to 4.2 million peptides, and all of that total will be 5.3 million. But if we are adding SNPs inside these peptides, and then you actually get a lot more combination. But when we add the SNPs, we don't actually use these exon-exon combination hypothetical cases. So total number of peptide covered is 7.8 million. So that's a lot more than what is actually recorded in today's protein databases. And that's where the high-performance computing component may come in. And you may actually say, well, this is really the layman's way of introducing the high-performance computing, but we thought this is actually helpful. So we're using the Quarry system at IU, and it's got maybe about 140 blade nodes, and got some, very marginal memory, and it's a lot better than a desktop computer, right? So what we did was we set up this experiment, and then this Zen server is where we store the raw proteomics data file. We actually subject this to both the local computer. It's actually a dual-core computer where we run this analysis in comparison with the one that we run on Quarry system. And then we also do some statistics outside of the system, and then we compare the results. And this is the performance that we got. And so for IPI, this is the standard database used for mass spectrometry searches. And this is the performance in terms of elapsed time. So when we use this very naive implementation of the high-performance computing, we get about 5-fold reduction in time. Not as much as we wanted, but it's improvement. And this database is only 47 megabytes in size. When we use PEPPI, just a knowledge base, meaning doesn't contain the theoretical peptides, and it's bigger in size, and the time performance actually doesn't actually still much better than just as database size double because of indexing introduced. And then when we don't have the SNP data, this becomes much bigger, and then with the SNP data becomes much bigger, but the time performance for local computer is a lot higher. So imagine, well, even though you may think, well, this is only maybe about an hour and a half, but for biology proteomics analysis that requires a lot of interactions, and only proteomics is only a minor step of so many steps of analysis, I would say that every improvement actually counts, right? And this is some actually result we run using a human fetal liver proteomics data that we pulled from the public repository of proteomics data sets. We actually used this database and used this framework. We found actually quite a few novel peptides that are quite new. So for example, this peptide, by the way, these are all passing a new statistical filter, so they're statistically valid. So we found this specific peptide actually existed because it actually included part of the introns. And this is quite amazing because this peptide have never been discovered before. And this part also have part of introns at the beginning. And so this one does contains a SNP inside this peptide regions. And this one also is exon-exon theoretical peptide. That means there's this exon skipping event that happens. So this is all the peptide that we discovered in all the proteomics data. Obviously, it highlights the problem we have in the proteomics experiment, which is we see a lot of false negative, and there are lots of peptides that may actually existed, but we couldn't see them because of the sensitivity of the instrument. But nonetheless, this is very encouraging because we're discovering something new because of this database we use and the HPC system that we put into the place. And another case study is that we actually run it on a breast cancer case. We take the blood samples of the breast cancer patients and the normal individuals. We actually clustered on about 27 protein isoforms discovered from our platform. And when we do the clustering, you can see that the breast cancer blood samples are significantly enriched with certain type of protein isoform, while the control are enriched in certain other type of protein isoform. So this is the first case that we've ever seen, and we haven't seen that report before, whereas the protein isoform has been used as a way of clustering and potential diagnostic applications for breast cancer diagnostics. So we're very optimistic about this approach, and we think there are lots of follow-up science that we can do that would benefit from this exercise. With that, and I would like to thank certain members of the group who are involved in this and DOD for partially funding this research. Thank you.
Thank you very much, Jake. Questions? Yes.
Yeah.
I was just wondering if you could talk about the algorithm or however you used to create just the peptide subunits from the original data.
Yeah, so there's a paper that we just published at BMC Bioinformatics, and then a lot of the details is in that. But essentially, we take the Ensemble database, we take a look at all the gene structures, and then we actually translate them into peptides. And for the junction peptide, we don't translate the whole region, but then we translate up to 240 base pairs, half of that from one the axon and half of that from another axon. And then we make sure that we're doing the translation in the correct reading frame and so on and so forth.
Thank you.
So thank you for the good talk. I just want to know, for the novel isoforms and SNPs that you discovered, did you validate that either with sequencing the SNP itself in the genomic sequence or cDNA sequencing?
Yeah, so we would like to validate it, and this is so new, so we're still looking for experimental biology collaborators. Yeah.
Somewhere between a question and a discussion point. A lot of what you're doing at generating theoretical isoforms reminded me of what you have to do if you're doing sequencing-based gene expression studies, because now your reads go across the splice junctions and you need to be able to recognise the splice junctions. And it would seem that if you were including that type of information, that would give you another dimension. You need to know all those theoretical forms anyway for the next generation of gene expression studies, and now you could link in, if you combined this work with sequencing-based gene expression, you'd have an even better way of determining what isoforms were there.
Right, exactly. So that's an excellent point. So we were just about to do some collaborations and for this type of a next-gen sequencing data. But initially, we were looking at the FE spliced junction chip, and they don't seem to have any useful data produced. The challenge here is that you have to find the right samples, and then the same sample is split for doing the proteomics and the next-gen sequencing project. So I think unless everything is perfect, then even if you get the next-gen sequencing data, you can just say, well, it's possible to have this, but is it really the case that they occur in the same biological context? So that probably would require another NIH study to figure out.
Okay, time for one more question.
Just a question in regards to the HPC environment you set up. Like, can you kind of talk a little bit about the throughput and the actual utilization of it? Because you said you had excuse me, 140 compute nodes.
Right.
And just kind of talk about that.
Probably Jeffrey would know better what the query system is about. So we have this query system, which is really not a best of what Indiana University has to offer, but it's an experimental cluster, and that's for us to crash and burn. So is this what you're asking about?
Kind of what was the throughput that you guys were actually kind of putting through this, and were you actually really fully utilizing the 140 nodes?
Probably not, but we were just pushing it to that system as a batch job, and I don't actually know why we're only getting a 5-fold performance increase than our dual-core desktop computer. We're still scratching our heads on why that's the case. Maybe it's because of the most of the time is not spent on the CPUs, and it's in just indexing and dispatching the job.
How long does each job take to run?
So here is about 800 seconds on the desktop, and then.
Yeah, but this must have many tasks. How long does each task take?
I have to find out.
My guess is that you may have too short a task, such as the other. In other applications, we have found that you need to get use, whether you're using Condor or whether you're using it, doesn't work well, and there's a backup tasks type problem because of what you said, the startup time is too great. But there is another obvious technology which will remove that startup time.
Okay.
We can talk when we get back.
Okay. All right. Thank you.
Thank you very much. I think we move on to the next talk. Okay, so we're ready for the second and final talk of this session. I'd like to introduce you to Paul Greenfield, who works for CSIRO, the Australia National Research Organisation. I'm afraid I don't exactly know what CSIRO stands for.
Scientific and Industrial Research Organisation.
There you go, that's the one. And he'll be talking to us today about querying genomes, answering biological questions by querying k-Mer databases.
Now you can hear me perfectly well. I'm echoing around the room. Good. Okay. We'll start with the first slide. Good. Okay. Some motivations for this work. It really started a few years ago listening to Jim Gray talk about astronomical databases. And that was really cool. You could actually answer real astronomical problems by just doing SQL, using some of the power of an SQL database. And at that stage, I was just getting involved in the biological side. I'm predominantly a database distributed computing person. I was just getting involved in biology, and I thought that'd be really nice to be able to do that in genomics. So that's what I was trying to do. That was the motivation of this experiment. And if I can answer biological questions with SQL, I can start doing ad hoc queries. I might have some data, 2 organisms, and I say, what have they got in common? Can I answer that type of question without having to write code? I might get some metagenomic data, and I'd like to be able to peer into the community and understand what genes are functional, what organisms are present, what variants of what organisms are present. Typical types of bioinformatics problems, but can I do it without having to write Perl scripts? The other motivation was, can I use the power and sophistication of a database to help me? Database engines are pretty good things. There were lots of performance optimizations built in, and tuned over decades, can I just make use of those to improve the performance of the tasks I want to do? They've got query optimizers, they've got inbuilt parallelism, they do much better buffering than I can do. They will do sequential read ahead when they detect that's happening. Lots of really good things. So to do that, I just need two things, or really one thing, a suitable database, which means I need, if I'm worrying about, I'm effectively comparing things up there, so I need some type of suitable similarity metric that I can drop in, and an appropriate set of organisms. So trying to do it on all primates would be really interesting, but there aren't that many primates in the databases. So all this work is founded on an observation or an assertion, depending how you want to put it. These are going to be k-Mer databases. A k-Mer is simply a short DNA string of length k. So that there is a 25-mer, and I mostly work with 25-mers. And some assertions. For a suitable value of k, and let's assume k is 25 just now, any given k-Mer will normally appear only once in an organism's genome. And if I look at all the, say, bacteria is what I'm working in, then if I've got organisms sharing k-Mers, then they will be related in general. If I share a lot of them, they're going to be very close normally. So close related organisms share many k-Mers, and those k-Mers that I do get shared imply shared function. So there's some assertions. In practice, 25 to 20, somewhere around there is about the right value for k. It's a good balance between the uniqueness and avoiding some of the problems that you get with accidental breakages as k gets bigger and bigger. If I made k 50, there'd be very little sharing because the chances of an evolutionary change would be too much. So, a related fact here is that some of my colleagues have been looking at the edit distance, Hamming distances of k-Mers about the same type of size. And for 25-mers, they're looking in plants and humans, not bacteria. You tend to need, adjacent k-Mers are about, if I have a k-Mer, then the nearest k-Mer to it in k-Mer space is about 8 to 10 changes away. So 1 or 2 changes, that doesn't happen. K-Mers are not very close to each other. It also helps the k-Mer space can be big. The 25-mer space is about 10 to the 15 possible 25-mers, and it's sparsely occupied. It's not sort of, everything's not down in one tiny little corner of it. They're just spread out over it. So there's some sort of pseudo facts to back that up. There's a bacteria looking at the distribution of numbers of copies of 25, and you see almost 99% of the k-Mers in that bacteria only appear once. The graph on the right-hand side is looking at the Hamming distance for Arabidopsis, and you see there we've got for about 25, these are for 36-mers, so we've got about almost 20, 25 average distance there. And I've also got some data based on a little spreadsheet. So I'll actually show you the spreadsheet if I had time. I might get time to look at that. The part of this work, I went and did a comparison of all bacteria. Easy enough. There's only 1,121 of them that I grabbed out of NCBI. And what this chart shows you is the number of 25-mers shared by the Escherichia, the E. coli and its friends. I did this particularly after listening to Vince's talk. So this bottom line here is Ferguson I, your favourite bug. And you can see, well, these are colour-coded. Of course, various shades of green mean various shades of sharing. And you can see there's a diagonal there, and self versus self is 100%, which is good. And as sometimes we have very close strains, and they're 95% similar. And typically, as you can all read this very easily, most of these strains are 40, 50, 70% sharing. So it's not too bad. That's just for the Escherichia. They're all strains, effectively. That's what the picture looks like in general. This is zoomed out a bit, and that's probably the might be archaea up in the top corner, and then some group like the firmicutes. And what we see in the general picture is that most bacteria share no k-Mers, share effectively none. This chart is done by me removing the ribosome, so otherwise you get this sort of background sort of sharing. Take out the ribosome and I get this type of picture. Most of the matrix is blank. There is no sharing, but nicely, when I first do this, I've drawn boxes around taxonomic groups here, but you can see the shadows of the major taxonomic groupings. Often you'll see, oh, there's a clump there, and that's a particular order. The taxonomy shows up quite nicely. And we also have something I've spent some time looking at and haven't finished looking at yet, really intriguing shadows. There'll be an organism's space up here, and down here somewhere will be a faint shadow of another group. So it's implying some type of shared operon by the look of my current analysis. So more work on that sometime. So, so what's in those little shadows? Well, here I just went and said, so if I take that Campylobacter and that Helicobacter, they're quite unrelated organisms, and say, well, gee, they share a very few 25-mers. What do they actually share? Where do those sharings come from? And it turns out that even though they're only sharing 2, 4, 1, 2, 5, 25-mers here, the gene annotation for what's being shared on both sides is identical, or mostly identical. So this is implying that even if I've only got just a very few 25-mers, they represent highly conserved critical parts of those genes. So let's talk a bit about a database. So I've got a k-Mer database. I take the genomes of every bacteria. I'm working in bacteria because there's lots of sequences available, and they're slightly less complex than eukaryotes. So I take every genome and I break it into a complete set of 25-mers. They're overlapping, I tile it. So if there's a 5 million base genome, then I get about 5 million 25-mers from it. And I drop that into a database table indexed by the 25-mer. I'm using shared k-Mers as a similarity metric, and which means that I'm going to run a program I'll talk about in a minute where I just cross-match. I count how many 25-mers from each organism I share with every other organism. So that's easy enough to do. And I drop that in as well. I do that count out of genome to genome, genome onto individual target genes, and source gene onto target gene level. So those tables. And there's lots of metadata in there about things as well. That complete database for this many bacteria and strains is about 620 gig. And that's fairly sloppy as well because I'm keeping all the genes there in ATCG form as well because it's useful to drag them out. And I keep all these k-Mers both in text form and in a 2-bit binary compressed form because it's convenient for me to do so. So if I really push that down, I could probably cut a couple of 100 gigabytes off that. And a lot of what I do is exact k-Mer matching. Exact k-Mer matching can be fairly fast. I have two ways of doing it just now. I either do direct queries on the index, and I recently got about half a terabyte of solid-state disk, and with a bit of a lot of careful coding and database structure, I'm currently doing about 200,000 lookups a second on that. And I can probably get a bit faster than that with a bit more tuning. And that's really good for what I would call sparse applications. A lot of my applications, I've got this very big database out here, and I'm touching that k-Mer and that one and that one and that one. So I may only be touching a fraction of a percent of the entire set. And so that's my sparse set. I've got other applications which are dense lookups. If I'm comparing everything with everything, then every k-Mer is going to be touched. And for that, the fastest way I have of doing it just now is just sucking the entire index into memory in partitions. I drop it into a hash table and then just mine it. And that gives me 500,000 or so lookups a second in aggregate. But most of that time is memory allocation time in the load, and I haven't even started trying to tune that. But that's not a very common application. I just do this when I'm populating my database, whereas I do this in production all the time. Why it's important to do. And that's plenty fast enough for my current purposes. I can cross-compare all the reference sequences in, well, if I had enough memory, it'd be about 2 hours on an ordinary quad-core workstation. That's fine for my purposes. Actually, in practice, it takes me more like a day because I can't actually keep all the counters. You know, this hits that type of counter. All those little ints just take up too much memory, so I've got to partition it and do it in multiple passes, but that's just memory. And a lot of what I'm doing is taking real read data from various sources, and that runs at this speed. And yeah, it's currently taking 10 to 15 minutes to run that through, which I'm quite happy with. It's not too long at all. And that's not worrying about porting to something large and grunty. So I said when I'm populating the database, I do this all versus all comparison. I simply generate all the 25mers, cross-compare them, and count various interesting things when we get matches. Pretty straightforward. Practicalities, I've got to look up about 4 billion bases, and I actually need to look up 8 billion because I've got to worry about the reverse complements as well. So I'm effectively doing 8 billion lookups in that time. That's it. Theoretically, well, not even theoretically, if I do the minimum possible counting, I can do it in one pass, and it's about 2 hours, which is okay. But to do that, there's pre-computed indexes, a lot of work on keeping threads synchronized so that I'm keeping track of working sets. But the nice thing about using the index is that I'm looking up this 25mer against all target genomes at once. Each single query does a one-to-many lookup. So I'm not really doing 8 billion times 4 billion lookups. That would take a little while. Now, I showed you a couple of tables before, the Campylobacter versus the Helicobacter. Where did that data come from? And it came from a query, from an ordinary SQL query. I've run that comparison. I've generated some data, loaded into tables, and then I can now do queries on it. And those numbers came from that query. And they're the types of queries I might want to do. I'll show you some of those. A nice simple query. I'm interested in the Yersinia, the Black Death bug, and it's in the Enterobacteriales order, and I want to find out what similarities there, what genes are highly conserved in that family, taking Yersinia as the thing, and that does it. I just go and run that query, and I get some answers. There's a small part of the answers. So the table's a bit longer than that, so I just took a bit of it. And so Citrobacter is one of the Enterobacteriales, and it says it shares these genes with Yersinia, which is interesting enough. They happen to be Yersinia-bactin genes, which is quite cute. So I didn't know what was being shared. I do that query, and boom, out pops these things. And what's often very interesting is these highly conserved hypothetical proteins keep on popping up as well. So one possible query would be, what are the hypotheticals? I've got a hypothetical gene here. I know that 25mers can be highly conserved. Perhaps I can use that to find a clue about what a hypothetical gene is. So I've got a strain of Lactococcus lactis, the cheese-making bacteria, and I'm saying go and look at all other bacteria and see if you can find places where a gene is hypothetical in Lactococcus lactis Chromoris SK11, but has an annotation in something else. And that's what that query does. And what's cute here is we've got some genes here which are hypothetical in our source bug, but are mapped to ABC transporter permeases in other lactococcus. So perhaps those hypotheticals are really these things. Not a bad way of starting to do that exploration. So you see quite a lot of these hypotheticals matching to things, or sometimes they're just hypotheticals match to hypotheticals and highly conserved, which is probably interesting in its own this is about as complicated SQL as I can write. And I'm not going to show you answers from this one. So I'm interested in finding unique genes. So I've got Citrobacter. One of my colleagues' wife works for a company which is interested in food contamination. So they want to know how to distinguish Citrobacter from other Enterobacteriales, like E. coli. And so which genes in Citrobacter are unique, are not shared in any significant way with the other Enterobacteriales. That's what that query does. Go and find all the genes in Citrobacter and subtract from that set all those genes which have more than 1% of shared 25ness. And that's what that query does. Now, that's all good. In fact, why don't I just go to here. There we go. As Vince is here, after your talk on Wednesday, I thought, oh, gee, have I got Ferguson IE in my database? Yes, I do. So what I did here was say, go and do a map from Ferguson I to one of the E. coli's and show me what's similar. And that's what that query does. In fact, it's a bit more sophisticated than that because I've got species ID in here. That's fine. Well, species ID there. I could make that family ID or order ID. I can go and do this comparison against some taxonomic level. So all I've got to do if I wanted to compare Ferguson I against all the Enterobacteriales or against all the other Escherichia is just change that part of the query and it'll run exactly the same. So that query works just fine and I get effectively a map out of it of what's in common between the Ferguson I and that particular E. coli. And as you can see down there in the fine print, that query took 8 seconds to run against all bacteria, against the all bacteria database on this laptop using that database. So performance is okay. And I'll just show you this as well.
Okay. Question before we move on to
yeah. Your writer's chair.
Yeah, I just wanted to ask you a quick question on that point before we move on.
Mm-hmm.
The performance, you say on that laptop with that disk.
Yeah.
Is that the solid state disk and
No.
what sort of capabilities?
No, that's just a 500 gig ordinary disk.
Okay, and this is SQL Server?
Yep,
and,
SQL Server, C# code,
right.
generating it.
What's the capability of your laptop?
It's a dual core 2.4 or something.
Okay, so kind of an average kind
Yeah,
of machine.
average type laptop.
Okay, I assumed that was with your special solid
No.
state. Okay, thank you.
Okay. And the last thing I'll just show here is so I don't do visualizations. I use Excel. And this is just me taking that data from that query you saw, dropping into Excel, color coding the % shared, and then also just mapping that % sharedness across the genome. And you can see that's what we've got. We've got some areas of high sharing. We've got some areas here which are quite different. I don't know what they are yet. It's easy to go and find out. So let's go back to the presentation as I'm winding up. Okay, so that's all stuff with reference data. And I think that's really cute, and I enjoy playing with it, and it's always full of interesting surprises. But my main job is actually doing work on real sequence data and lots of applications of that. Do the same thing, get a batch of sequence reads, turn into k-Mers, throw the k-Mers at the database and see where they stick, and then do interesting things after you found out where they stick. That runs in the sparse matching time. It's about 10 to 15 minutes these days. And I'm interested in questions, well, I've got someone's got cultured a bug and they've got some sequence data from it. How different is it? What SNPs are there? What genes are missing? What new genes are there? Those types of questions I'm interested in. And increasingly these days, metagenomic applications. We go and take an environmental sample, oil, water, coal, and do the same thing with that. And then find out what's in there And yeah, there's metagenomic applications. So here's some outcomes from that. I've got a very similar database for 16S. So I can take the metagenomic reads, complete set. I can filter them into ones that include bits of 16S and don't, using 25ers. That works very well. So now I've got a collection of 16S only reads, the reads that contain fragments of 16S. And then I can throw that against a 16S k-Mer database and do some counts. And these pie charts there do that. Pretty fast to do that. And those numbers are pretty good compared to other ways of determining metagenomic communities. So the more traditional PCR and Sanger sequencing based ways. So that's that. The top here says, let's partition the reads. Now if I do the reads, I say, gee, I'm getting 20% of my reads are from the Desulfo Vibrio and AC, which is good, we've got a Desulfo Vibrio in there. But you see we run out of, it doesn't add up to anything like 100, because I can only give you information about bugs that are close to ones in the NCBI database. And when I do a real metagenomic environmental sample, there's lots of novel things in there. So that's really useful, and I can sort of partition things out, but there'll always be a big others. In this particular sample, we've got a Methanococcus, which is novel. And part of the job is going to be sequencing that properly. When I do that run against, splattered against the database, one outcome of that is a set of these mapping tables for every known bug. This file pops out, and so this is a bit from the Desulfo Vibrio. So it says, well, here's what the metagenomic reads say maps onto the Desulfo Vibrio reference organism. So I get this sort of heat map type thing of every gene in the reference Desulfo Vibrio. And I do that in one run for every reference bacteria. Okay, finishing up, problems, extensions, the elegance of the matching code. I'd really like to get the database to do very simple matches even faster than it does. And everything I tried to do it faster, more sophisticated, ends up being slower. So that would be nice sometime. This application is embarrassingly parallel and all that type of stuff, so it'd be really easy to run it on a cluster because the lookups are all separate. And more biology being added into the database. I've currently got COGS groups in there for protein function or gene function. I haven't played with much that, but it's in there. And sometime real soon I'll be putting in the amino acid codings rather than just, so if there's been synonymous changes at a codon level, that will be fine. So that'll make some things that look a little bit different, a little bit closer. And I'll probably do something about simple close matching, worrying about one-base substitutions, etc. That might require some different index structures. And there's some work going on as well about automatically finding SNPs based on, again, on k-Mer approaches. So there we go. There's a summary slide which I won't bother reading out. And I'll hand over to Simon.
Okay, are there any questions?
Yeah, so I can see the good values in using database to do this type of work. So I have two questions. One is that there's a BLAT program from by UC Santa Cruz, and that can achieve pretty much the same functionality. And of course, it doesn't run inside a database environment. I wonder if you have a look at that.
I use BLAST all the time.
BLAT, B-L-A-T.
Yeah, BLAT, yeah,
okay.
Well, that's the same. Yes, I use those type of things all the time, mostly as verification that what I'm doing is right. And one nice, I said I do the partitioning of the metagenomic data. One of the really nice things about metagenomic sequence data is that every read is an individual, and you're getting samples down at very rare organisms. And even the ones where I say, this belongs to something, and I'm only seeing five reads from it, and I'll take those and blast them back against the references, and they pop up. But you said you were comparing two organisms using NCBI BLAST and sort of 12 hours or so overnight to do it? So there's several orders of magnitude difference in speed. The problem. This is an exploratory tool rather than exactly how do these things. If I did what Vince had done and had that display, then it would be nice to click on a box and then do a blast of the two genes. So I don't see it as being an alternative to BLAST and its friends. I see it as being a complement to it.
So a second question is regarding the performance. So in the example you showed, just mapping one 25-mer was 8 seconds. So for any metagenomics application, you have up to 1 million of that.
No, that was comparing two organisms in 8 seconds.
Okay.
The typical run of taking a set of metagenomic reads, saying a gigabase of 454 data, is about, against the entire database, is about 10 to 15 minutes.
Okay. All right.
So thank you again for the excellent talk. I can see, I've been thinking about something similar to this for a while, and I never really had the expertise to actually pull it off. So I can already see about 5 or 6 other applications that you can do with this. So just by even comparing samples to each other, so different metagenomic samples, and rapidly seeing whether how much they share among each other versus how much they're different, and whatnot. So how do you see this being deployed? And
well, someone could host the database publicly. That would be nice. I can give you a copy of the database. So depends on what you want to do. If you want to build applications around it, then it's small enough you just host it yourself. So I said the entire database was 620 gig with a lot of wasted space in there. The database I'm doing those queries on doesn't include the k-Mers because they're not interesting for me doing the queries. And that database is 20 gig. So the functional database for a lot of the work is 20 gig. But if I'm doing the sequence data matching, it's a lot bigger. But so people could either host, particularly the small one, publicly for doing queries, and then for the larger one, you'd probably have your own copy of, and just use it.
And so SQL Server free, or?
It uses current SQL Server. No problem.
You said it's SQL Server free. No, it's a product. But there is a version called there's the Express edition, which is for certain groups free. You'd have to read the license.
Yeah, except Express has limits on table sizes.
Oh, okay. So no, it's not free. I was thinking, since you benefit, as you say, from parallelism, or you have an embarrassingly parallel problem, you might want to take a look at something like the Azure database.
DB,
whatever it is.
Those things are all on the list to look at.
Although, to be honest, scalability doesn't seem to be your problem because performance is very rapid anyway.
Yeah.
So, you're not necessarily going to win.
No, there hasn't been enough pain in the performance to make me go and look at those software engineering tasks of putting more. It's obviously highly threaded code already,
But 3 to 5 years from now, there's not going to be 1,000 bacterial genomes out there.
Oh, no.
There's going to be insane numbers.
Yeah.
Yes. Anyway, enough from me.
I just had one quick question. You talked earlier about having to do the reverse complement of the k-Mer.
Yeah.
And is that just because you're not convinced on whether or not you're working with forward or reverse strands? Of
Oh yeah,
bacteria?
if I'm dealing with sequence data, then I don't know which strand the read came from. If I'm dealing with reference organisms, then there can be translocations, and you can swap over the other strands. So I don't know where it came from.
It's unfortunate because you got to double.
Oh yes,
yeah,
yes, yeah. It's only a factor of 2.
Are there any other questions? Oh, one more.
Yeah, Paul, it's very nice. I was interested in the fact that you got, you were able to use the 25-mers as a kind of a nice signature. I've seen those plots before, and they're still very impressive. What I was interested in, though, is whether you've found anything in the sort of 25-mer signature of an organism in the non-coding regions. Because most of the stuff that you're getting there is sort of conservation in the coding sequences.
Yes, but most of those queries say and PID not equals to non-coding. So
Right, okay.
there is lots of conservation in the non-coding region.
So I mean that's really interesting. So,
yes, given the work you were doing on regulation, yes.
Yeah, exactly.
It's highly, the two things that are really interesting are highly conserved non-codings and highly conserved hypotheticals. And they both just stick out.
Okay, we'll talk further then.
Yeah.
Okay, well, we're running a little late. I believe the panel session starts in 15 minutes downstairs in the Empire Ballroom, so we'd better draw this to a close. There's coffee outside. Please join me in thanking the speaker.